This is Athar Malik presenting a video presentation on therapeutic strategies for repair and regeneration following traumatic brain injury by Athar Malik and Mark Richardson from the Department of Neurosurgery at Harvard Medical School and Massachusetts General Hospital. By the end of this video, viewers should have a better understanding of the principles of recovery following traumatic brain injury and the potential therapeutic strategies for recovery following traumatic brain injury. When we think about the idea of repair or regeneration following traumatic brain injury, it is notable that this is a relatively recent idea. Less than 1 century ago, it was believed that the nervous system was incapable of repair or regeneration following injury. Nobel Prize-winning neuroscientist and neuroanatomist Santiago Ramon y Cajal summarized these sentiments in writing, quote, "In adult centers, the nerve paths are something fixed, ended, immutable. Everything may die, nothing may be regenerated. It is for the science of the future to change, if possible, this harsh decree." End quote. Since Cajal's time, a large body of scientific research has fundamentally changed this understanding, leading to new opportunities for neurosurgical treatments of traumatic brain injury. Historically, neurosurgical treatments for TBI have focused on interventions during the acute phase of injury to minimize the primary injury and prevent further damage from secondary injuries. These traditional strategies are shown with orange arrows, and they focus on reducing sequelae of the primary brain insult to salvage acutely threatened tissue. Now, there is increasing interest in the TBI community to enhance the nervous system's endogenous capacity for repair, regeneration, and functional recovery. These restorative strategies, which are shown with blue arrows, represent a shift in therapeutic goals from focusing solely on protecting acutely threatened tissue to introducing interventions that support spontaneous and directed functional recovery. Much of the evidence for spontaneous recovery of the damaged cerebrum comes from the stroke literature. Animal studies have provided cellular and molecular information, and human neuroimaging and neurophysiology studies have provided system-level insights. Clinically, several basic principles of recovery have been identified. 1. Most spontaneous recovery typically occurs within 3 to 6 months of the injury. 2. Cognitive deficits are more likely than motor deficits to show further gain beyond this point. 3. The rate of recovery is inversely proportional to the severity of the deficit. And 4, recovery patterns vary between different types of deficits. These observations suggest multiple context-dependent mechanisms of recovery that can be generalized into 3 basic categories: plasticity of intact networks, repair of damaged circuitry, and replacement of lost neurons. This presentation will describe the opportunities and obstacles inherent in potential therapeutic strategies for repair and recovery after TBI, including manipulation of endogenous plasticity and neurogenesis, cell transplantation, gene therapy, neurorehabilitation, and electrophysiological manipulation. These approaches may be used to facilitate recovery or functional reconstruction in several neuroanatomical contexts. The neuronal loss in traumatic brain injury occurs across multiple anatomical and physiological regions and involves the interruption of multiple brain circuits with a varying severity. Opportunities for initiating potential restorative therapies are therefore present at these multiple neuroanatomical sites. For example, mobilization of subventricular zone neural progenitor cells, ipsilateral and contralateral to the injury, salvage and replacement of damaged neurons in the vicinity of the injury, and mobilization of dentate gyrus neural progenitor cells, ipsilateral and contralateral to the injury, as well as peri-injury plasticity and plasticity in more distant brain regions, in this case contralateral to the injury site. At these various neuroanatomical sites, in the setting of traumatic brain injury, complex changes occur in the cellular microenvironment as a result of both pathological and recovery mechanisms. These changes involve inflammation, reactive gliosis, disruption of physical structures and protein expression in the extracellular matrix, altered expression of trophic factors, and neurogenesis. Although some of these mechanisms attenuate acute damage at the expense of future regenerative capacity, and thus play a disabling role, others facilitate recovery and thus play an enabling role. Modulating these responses may provide a means to promote functional recovery. Plasticity is defined as the ability of the nervous system to adapt to external stimuli, and this includes adaptations to injury. While plasticity occurs when injury induces reorganization of cortical connections, it is not always synonymous with functional recovery. Even though processes such as synaptic sprouting, unmasking of dormant circuits, and the development of new synaptic connections can facilitate recovery, plasticity can also result in abnormal function, such as the development of post-traumatic epilepsy seizure foci, or post-traumatic neuropathic pain. Modulating plasticity responses may provide a means to promote functional recovery. Additionally, traumatic brain injury induces changes in the specialized microenvironment that regulates the proliferation of neural stem cells and the differentiation of neural progenitor cells. These neurogenic niches are found in the brain, in the human subventricular zone of the lateral ventricular walls, and the dentate gyrus of the hippocampus, as shown here. Animal models investigating TBI-induced neurogenesis have shown that the numbers of neural progenitor cells in both the subventricular zone and the dentate gyrus are substantially increased after TBI, and this process is more robust in juvenile animals. This figure illustrates the increased cell proliferation in neurogenic regions after experimental TBI. In panel B, we see an increase in the number of bromodeoxyuridine BRDU labeled cells in brown in the ipsilateral dentate gyrus of lateral fluid percussion injured rats 2 days after injury as compared to sham animals shown in panel A. Similarly, in panel D, we see BRDU labeling in the ipsilateral subventricular zone of injured animals is significantly increased compared to sham animals shown in panel C. Newborn neural progenitor cells from the subventricular zone appear to have the potential to migrate to areas of focal cortical damage, whereas hippocampal neurogenesis occurs in the setting of diffuse local damage. In addition to attempting to promote the beneficial effects of these endogenous cell populations, an additional method that has been proposed to promote recovery after TBI is cell transplantation. The clinical trials undertaken thus far involving cell transplantation into the brain have been for patients with Parkinson's disease and Huntington's disease. It is important to note that there are critical differences between cell grafting in the striatum for Parkinson's disease, for example, and the cell grafting that might be useful for patients with TBI. One of the putative targets for TBI, for example, is the hippocampus, and it is a neurogenic niche itself, and therefore may be more likely to permit the survival and appropriate differentiation of transplanted neural progenitor cells. In addition, trauma-related factors that alter the integration of adult newborn neurons into the hippocampal circuitry will also be relevant for the integration of transplanted neural progenitor cells. In contrast, focally damaged areas of the neocortex offer a less welcoming milieu for the survival of transplanted neuronal cells, and transplanted cells may be less likely to survive in this hostile environment acutely after TBI. Strategies for clinical transplantation in TBI will probably vary with patients' individual anatomic pathology. Graft locations will also be tied to the desired functional role of the grafted cells. Replacement neurons, remyelinating oligodendrocytes, and supporting chaperone cells, for example, are all potentially useful cell types whose relative value may change depending on the clinical situation and the precise anatomical site of transplantation. There are several possible cell sources available for cell transplantation. These include neural stem cells, neural progenitor cells, autologous neural progenitor cells, induced pluripotent stem cells, oligodendrocyte precursor cells, and mesenchymal stem cells. When thinking about how to optimize transplanted cells, several studies have shown that biological scaffolds may play a critical role in optimizing the microenvironment surrounding transplanted cells to facilitate neurogenesis, migration, and proliferation. Regarding the possible cell sources for cell transplantation, because of issues of immunosuppression, allogenicity, availability, and potential neoplasia related to the use of neural stem cells from other sources, autologous neural progenitor cells would be an attractive source of cells for first attempts to repopulate the injured brain at sites of focal damage or within the hippocampus. As an initial model, subventricular zone neural progenitor cells from the adult rat have been expanded in culture and allogenically transplanted to the adult rat hippocampus to test their ability to transdifferentiate to neurons appropriate to the hippocampal niche. This figure shows a rodent model of adult neural progenitor cell transplantation. Neural progenitor cells may be isolated from the adult subventricular zone as shown in panel A, and subsequently propagated and manipulated in cell culture, as is shown in panel B. They can subsequently be transplanted into the adult hippocampus, shown in panel C. In the dentate gyrus, which is shown with a black square in panel C and zoomed in panel D, transplanted cells are shown in green, and we see that they disperse. However, only those cells in the subgranular zone, which is shown in panel D with the white square, and zoomed in panel E, only these cells express a hippocampal granule cell neuronal phenotype. By 2 weeks after transplantation, nearly 80% of the new cells that were located in the granule cell layer had the immunohistochemical profile of granule cell neurons, thus suggesting the ability of adult subventricular zone cells to transdifferentiate to locally appropriate neurons in the hippocampus. In a separate study, human neurosphere-derived neural progenitor cells have been transplanted into immunosuppressed rats that underwent fluid percussion injury. Both graft cell proliferation and more robust incorporation into the host hippocampus were seen after injury compared to uninjured animals. Surprisingly, human autologous transplantation of tissue containing putative neural progenitor cells harvested from contusion zones and reintroduced into TBI patients has also been reported, but further studies are needed to demonstrate the efficacy of these approaches. Now, in addition to cell transplantation methods, gene therapy for brain disorders is one of the most promising frontiers in the practice of restorative neurosurgery. There are significant experimental gene therapy initiatives underway that have led to currently active clinical trials involving the direct intracerebral delivery of viral vectors for treating neurodegenerative movement disorders, and these treatments have been reported to be safe and well tolerated. It is conceivable that many of the neuroplastic molecular interactions discussed earlier could be targeted for direct genetic manipulation. Patterns of gene expression that vary in injury severity and recovery time suggest that the pathophysiological cascade induced by TBI is accompanied by molecular responses that may offer windows for therapeutic interventions, including gene therapy. The blood-brain barrier prevents significant amounts of most systemically administered agents from reaching therapeutic levels within brain parenchyma without producing peripheral toxicity. Initial attempts at direct local delivery of therapeutic agents into the brain have relied on diffusion, which resulted in non-homogeneous distribution restricted to a few millimeters from the source. In contrast, convection-enhanced delivery, or CED, uses a constant pressure gradient established at the tip of an infusion catheter to distribute the therapeutic agent by bulk flow. This method produces an even distribution of highly concentrated agent over considerable distances, the volume of which is dependent on the structural properties of the tissue and the parameters of the infusion procedure. Now, this method of convection-enhanced delivery in a traumatically injured brain presents a significant challenge because of disruption of the blood-brain barrier, white matter tracts, and perivascular and extracellular spaces. Convection-enhanced delivery has yet to be formally studied in experimental TBI models, and eventual clinical use will require intraoperative imaging techniques that provide real-time feedback on viral vector distribution. In addition to the previously mentioned methods for recovery following traumatic brain injury, an additional method is focused on neurorehabilitation and environmental enrichment. Injury-induced microenvironmental pathways are dynamic and responsive to stimulation-triggered external factors, such as physical activity and environmental enrichment. The clinical effects of stimulation-triggered neuroplasticity have been examined primarily at a behavioral level, but neurorehabilitation interventions based on this concept have shown some success. Several mechanisms have been proposed to underlie these functional and histological improvements following neurorehabilitation and environmental enrichment, and those are summarized in this figure. For example, BDNF is increased in the ipsilateral hippocampus in animals that have been exposed to environmental enrichment. Traumatic brain injured animals in environmentally enriched housing have increased dendritic density in ipsilateral occipital cortex, supporting the idea that environmental enrichment leads to behavioral improvement by enhancing plasticity. Indeed, increased hippocampal neurogenesis and increased number of neural stem cells have also been reported. In addition, genetic expression profiles of substantia nigra and ventral tegmental area are modulated by environmental enrichment in the setting of TBI. These changes include genes associated with calcium signaling, dopamine, and membrane homeostasis. Recent retrospective reports on the effect of environmental enrichment on outcomes after TBI have begun to demonstrate its benefit in human TBI. Environmental enrichment after TBI reduces atrophy in the hippocampus, whereas post-acute rehabilitation within 3 months of injury is associated with improved outcomes. In addition, several randomized clinical trials have been reported for environmental enrichment in humans following TBI, demonstrating functional improvements. There has been an increasing interest in various neurostimulation strategies to enhance functional recovery after traumatic brain injury. Of particular interest is a non-invasive group of techniques including transcranial magnetic stimulation and transcranial direct current stimulation, which have demonstrated effectiveness in several studies of stroke patients. The use of these techniques for traumatic brain injury has been less common, with only a few case reports showing TMS improving neurobehavioral function and TDCS improving attention. Although there are many mechanistic differences between the injury from stroke and that from TBI, one may postulate that application of these techniques in TBI may result in some of the benefits seen in stroke patients. The advantages of these non-invasive stimulation strategies include lack of any substantial recovery period and the ease of application. However, these techniques have several limitations that warrant consideration. It is difficult to target deeper structures using these techniques because the electrical current and magnetic fields generated are less predictable. Moreover, these are temporary stimulation techniques with treatment effects that may diminish months to years later. Direct stimulation of the cortex after surgical exposure has also been reported in the literature, both in humans as well as in animal models. In rats and primates, cortical stimulation with surface electrodes during the rehabilitation period resulted in improved motor function. After a period of rehabilitation and stimulation, there was an increase in the size of the cortical area representing movements, showing that electrical stimulation during rehabilitative training enhances development of new areas of movement representation. Deep brain stimulation has been widely popularized in the treatment of movement disorders such as essential tremor and Parkinson's disease. Because sequelae of TBI include post-traumatic tremor and dystonia, the use of DBS in the setting of TBI for symptomatic relief of movement pathology has been explored in several small case studies. Several studies have also demonstrated the use of DBS in enhancing cognitive function and arousal in TBI patients. Future studies are needed with randomized groups of patients who will receive DBS or no stimulation, as well as a longer follow-up for several years to determine long-term outcomes after stimulation. This slide summarizes some of the potential restorative therapies for TBI that we've discussed in this video. There is currently more speculation regarding the potential for repair and recovery after TBI than there are data describing these processes in the human brain. Most of us, however, have witnessed cases in which some severely injured brains do achieve meaningful functional repair against significant odds. As progress is made in improving functional neuroimaging techniques, this technology should be coupled with increasing knowledge of the neuroplastic mechanisms initiated by brain injury to improve prognosis and identify patients with potential for functional recovery. Identification of successful neuroplastic processes in patients will provide targets for developing therapeutic agents to augment these responses. Delivery of gene therapy by direct infusion of viral vectors will potentially be used to bolster endogenous neuroplastic processes pending identification of appropriate molecular targets. The degree of circuit integrity remaining after injury is probably a recovery limiting factor. DBS may help facilitate identification of these pathways as the mechanisms underlying the wide success of DBS in the treatment of movement disorders and limbic dysfunction are reapplied to post-traumatic conditions. Rebuilding neuronal circuits with regenerative therapies such as the mobilization of endogenous neural progenitor cells or transplantation of exogenous neural progenitor cells, or both, will depend on several unresolved issues, including cell source, phenotype, and ability to integrate within disrupted anatomic scaffolding. Choosing appropriate post-injury time windows for each of these specific interventions will be critical in determining their success. In summary, the brain has endogenous mechanisms that facilitate recovery after traumatic injury, in striking contrast to what was believed only a century ago. Mechanisms of recovery include plasticity of intact networks, repair of damaged circuitry, and replacement of lost neurons. Interventions that promote endogenous pathways of repair have shown promise in clinical trials. Potential strategies for facilitating recovery after traumatic brain injury include manipulation of endogenous plasticity and neurogenesis, cell transplantation, gene therapy, neurorehabilitation, and electrophysiological manipulation. Further work is needed to better understand endogenous mechanisms of recovery as well as possible interventions to improve outcomes after traumatic brain injury. Thank you for your attention, and for more information, please refer to our book chapter, Therapeutic Strategies for Repair and Regeneration Following Traumatic Brain Injury.